Our next panel is Unfinished Triumph, Universal Flu Vaccine Update. The universal flu vaccine shows promise in providing broader and more durable protection against both seasonal and pandemic influenza viruses. In this session, we'll discuss progress on the development of a universal flu vaccine or vaccines by taking a broad look at the overall landscape and then looking specifically at an NIH clinical trial and development. Our speakers include Dr. Michael Osterholm, Director of the Center for Infectious Disease Research and Policy at the University of Minnesota, as well as Dr. Matthew Mamoli, the Director of the Laboratory and Infectious Diseases Clinical Study Unit in NIH. Dr. Osterholm, I see you on Zoom. Your slides are up. You have the floor.
Thank you very much. It's a real honor to be with you here today and appreciate the chance to share with you work that we have been spearheading, but nonetheless have only been a small part of in terms of all the experts who have participated in this work. Today, I'm going to briefly summarize for you two different aspects of our work. One is the influenza vaccines R&D roadmap, a process that is still ongoing, but as you'll see in a moment, we've achieved some important milestones, and then an ongoing monitoring effort to understand what's happening with influenza vaccine technology development with the universal influenza vaccine technology landscape. If I have the next slide, please. So let's first start out with the IVR initiative, the influenza vaccine roadmap. What you see here is our homepage on our website, which is available to all, obviously. And on the bottom left, you see the roadmap. You can click that and actually get access to the roadmap materials that I'm going to cover today, as well as much additional information. And then on the right side, you'll see the influenza vaccine landscape. For which, again, the second part of what I just mentioned, you can get information there. If you look at the top up above, you get the same thing. It talks about the whole IVR initiative, how it's supported, how to get involved, how to provide information, our routine newsletter, and then extensive resources that are available to anyone who wants more information on influenza vaccines. Next slide. Now, about the IVR initiative itself, this was really all about documenting and moving forward new advances in vaccine science and technology that must be leveraged here, as we understand, to usher in the new era of influenza vaccines to better protect all of us. And the IVR provides a detailed roadmap for doing this. It was initiated in 2019 with support from the Global Funders Consortium for Universal Influenza Vaccine Development, initiated this whole process. If you look at the lower left, you can see in addition to CIDRAP, our center, the World Health Organization, the Sabin Foundation, the Bill and Melinda Gates, the Task Force for Global Health, Wellcome Trust, Rockefeller Foundation, and the Global Funders Consortium for Universal Influenza Vaccine have all been supporters of this activity. And during the period from 2019 and 2021, we had numerous meetings. Of course, in the world of COVID, many of those had to be on Zoom, but nonetheless, we had extensive involvement from experts around the world. And in 2021, the roadmap was actually launched and available. Now we're in a period since that time of 2022 with IVR monitoring, evaluation, and adjustment. Continuing to keep track of where the roadmap is at and what else we need to do with it. Next slide. The roadmap is a 10-year plan for prioritizing and coordinating global influenza vaccine R&D. We've identified a number of issues and barriers. We've identified 24 strategic goals, 113 milestones, 37 were designated as high priority, and then additional research priorities. There are 6 key topic areas with goals and milestones for each. Vaccinology for universal influenza vaccines, vaccinology for seasonal influenza vaccines, the overall virology, immunology, animal and human models, and policy, financing, and regulation. Really from soups to nuts. Next slide. About the landscape now, let me move on to that and share with you, this is a database of novel influenza candidates in clinical or late preclinical development. It's publicly accessible via the IVR website. It currently is funded by the Global Funders Consortium for Influenza Vaccine Development, and it is continuously updated as new information becomes available. The link you can see at the bottom of the slide here. Within this, we are monitoring and continuing to assess 115 vaccine candidates that are in preclinical or clinical development. We look at 28 vaccines in clinical trials themselves. We are looking at 123 different developers and including 6 different platforms. Again, if you go to the top nav bar, you can look at the landscape in terms of tracking and the information that's available. Next slide. As I noted before, we have the different groups here in terms of vaccine candidates, vaccine and clinical trials. If you look at the very bottom, you'll see here that in terms of novel vaccine candidates, we have 122 in preclinical, following 16 in phase 1, 8 in phase 2, 4 and 3 phase 3, and none have obviously been approved yet. You're going to hear about one of these in just a moment. Again, just to reemphasize, universal influenza vaccines are the novel vaccine candidates designed to provide broader and more durable protection against circulating pandemic influenza viruses compared with the current strain-specific seasonal influenza vaccines. So we surely distinguish between these two. Next slide. This is an array of the number of universal influenza vaccine candidates by platform and phase. And each one of these, as you'll see on the left vertical bar, the different types of platforms, and you can see for each one the number that are in each of these different phases. And our job is to keep this as current and as comprehensive as possible. Next slide. If you look at the universal influenza vaccine candidates in clinical development, we give you more information here by each of the platforms I just mentioned. Again, you're going to hear about one shortly, but whether it be the recombinant proteins, the recombinant influenza vaccine virus-based, virus-vectored, virus-like particles, non-VLP, non-nanoparticles, or nucleic acid-based vaccines. And these are some of the examples here of the candidates that we're talking about right now. Next slide. One that you're going to hear about here is the NIAID BPL 1357. Within our site, if you look down under phase 1 and key sources, you can click on any of those and get additional information about this specific vaccine, where it's at, what's happening in terms of its status. And this is something, again, as you're looking across all of these developers, we are making every effort to keep these current. Next slide. In terms of the universal vaccine technology landscape provides data for monitoring R&D progress towards improved influenza vaccines. We know where we're at that way. It's identifying strategies for future research based on the findings of what we've learned to date. It's informing R&D funding decisions and where we can see the need for additional support with priority areas, and then just analyzing the R&D funding trends. We obviously are all concerned about, in a time of COVID, the concentration that was necessarily important for dealing with COVID, but what would it do to other aspects of the infectious disease vaccine portfolio? And so we're obviously very keenly aware of that and keeping track with that. Again, this information is all available on the Universal Influenza Technology Landscape pages. Next slide. So if you want more information on this, you can contact our IVR team at ivr.umn.edu. If you want to visit the website, https://ivrsidrap.umn.edu. We are adding an interactive part of the site next year with the landscape area. So if you want more additional information, you'll be able to go in and interact with it. So I thank you again for the opportunity to provide this very brief overview. We hope that this is useful to you. We continue to express our appreciation to the supporters of this effort and believe that it does serve as a very important, you might say, stake in the ground to understand the current and hopefully the future status of influenza vaccines. So thank you very much.
Thank you, Dr. Osterholm. Our next presenter is Dr. Matthew Mamoli, the Director of the Laboratory of Infectious Diseases Clinical Study Unit at NIH. Dr. Mamoli, you have the floor.
Thank you. Thank you for inviting me today to talk to you a little bit about our work. So I'm going to present to you a little background information on the vaccine that I've been working on with Jeff Taubenberger at NIH for the past 10 years, and then talk to you about the clinical trial that we're very excited to start this year. So let me give you a little background information here. Am I using the right clicker? Nothing's happening when I click the button. So maybe if somebody can advance it for me. No, I don't. There's nothing here, just a little clicker on the side. Okay, that'll work. We'll just advance the slides. So the work that we're doing has come from really 16 years of research. I started out working with Jeff Taubenberger in the viral pathogenesis and evolution section at NIH about 16 years ago now. We built up a translational program together. He was focused primarily on the animal work, and I was focused primarily on the human work. And in conjunction between healthy volunteer human challenge studies and a lot of pathogenesis work in animals, we really came to some conclusions about how we need to approach a mucosal RNA virus like influenza. And as you're aware, these viruses present some unique difficulties, viruses like flu, paraflu, coronaviruses, where you not only have the issue of mutation and changing of the virus because of its RNA nature, but you also have issues with how they cause infection, and that they're causing infection primarily through a mucosal route. There's issues with tolerance, there's issues with getting immunity to the right place, and a whole host of other issues that have made it extremely difficult to develop vaccines that really do what we want them to do, which is not only reduce mortality, but also reduce infections, reduce transmission. And so up till this point, and after decades of working on influenza and now coronavirus, we still have not solved the riddle of how do we prevent infections, prevent transmission, and really intervene in a significant way. Even in our best year with influenza, we get a 50 to 60% effectiveness rate, and most years it's lower than that. Last year was 16%. So we really need to do better and find ways to do that. And so all of this work that we've been doing, which is sort of very dramatically summarized here on this slide, is the work that we did as a background to help inform us on how to make this vaccine. Next slide, please. So what was our goal with this vaccine? Well, our goal was to make a universal or broadly protective vaccine. And as you saw from Mike's slides, there are many ways to try to go about that. And there's many sort of definitions of what a universal vaccine for flu would look like. In our case, we want it to be as universal as possible. And so that not only means breadth of protection in terms of how many viruses can we intervene and protect against, or strains, but also breadth of protection in terms of the types of people we can protect. Can we protect the elderly? Can we protect the young? Can we protect people on disease-modifying agents with cancer and other underlying illnesses? We also want to have a breadth of protection in terms of the mechanisms of immune response and the mechanisms of protection offered. What we've learned in our studies is that when we focus on what everyone always focuses on, systemic antibody against a single antigen, we're actually only focusing on a tiny little component of the immune system that protects against these viruses. And through years of doing challenge studies, we've really identified that antibodies against the HA really are in the blood are really only predicting a tiny amount of protection, and that we need to look at other things. Some of those things may be mucosal, some may be cellular, but it's really a large immune response on the whole to more than one antigen that we really needed to focus on. And so that's what we're trying to do with this vaccine. So our hypothesis was that an inactivated whole virus vaccine presenting multiple antigens through an IM and an IN route would provide broad protection. Next slide, please. So the vaccine, the first candidate we came up with, we actually have two right now. This first one is called BPL-1357. So what is it? It's a vaccine cocktail of 4 low pathogenicity avian influenza viruses. And in fact, they're not recombinant at all. They're actually wild type flu viruses that were collected from birds in nature. Their whole virus is completely wild type. We took those viruses. We grew them under GMP conditions. The final GMP production is done in certified Vero cells that NIAID has. They're then inactivated with beta-propiolactone. They go through extensive sucrose gradient purification and safety testing. And then we had our product. And we make each virus individually, live, inactivate them, and then they're combined to make the vaccine. Next slide, please. So how did we pick this? Why did we do this? Well, so we started looking at phylogenetic trees, and you can think of these as sort of family trees of influenza viruses. And what we do is we made two family trees based on the two most variable parts of the flu virus, the hemagglutinin and the neuraminidase, HA and NA. And what you see on this slide, I circled the 4 HAs and the 4 NAs that are included in this vaccine. And as you can see, they're all off sort of different branches of the family tree, sort of spanning as much of that tree as we could when we use both the HAs and NAs. It's hard to see on the NA, but the little HAs are there that they go with. So just trust me, we tried to cover it as much as possible. And we actually have a second candidate that does the same thing. So we have 2 candidates that kind of try to cover the breadth of influenza virus strains. And these are avian, and all human flu, basically all flu in every other species, has originally come from these avian viruses. So these are sort of like great-grandparents to all influenza viruses that have jumped into mammals. And so the idea was, can we pick a broad array of viruses that represent these ancestors of all mammalian flu and use those as a vaccine? Next slide, please. So these are the viruses we picked for this one, an H1N9, an H3N8, an H5N1, and an H7N3. We went ahead and put 0.25 mils of each virus in our vaccine, giving us approximately 100 micrograms of total protein of those viruses. And so we end up with about 400 micrograms of total protein in each dose of the vaccine, which is in 1 ml. Next slide, please. So some quick preclinical data. We did some studies in mice initially, vaccinating the mice. We did lethal challenge with a number of different viruses, some which matched the hemagglutinin neuraminidase in the vaccine, others that did not. And we saw really great results. We were giving them 10x LD50s, or 10 to 50 times LD50s in some cases, of these viruses, and we saw 100% survival. Next slide, please. So as you can see here, so some of the matching HAs, the H1 up in the top left, and we have I think it's an H5 over there on the right. Those match. You can see 100% survival. Those are the ones with just the straight lines. Those are the survival. Whereas the MOX dropped off. We gave it IN and IM, and we saw really good results, in particular with the IN. If you look down at the H10N7 or the H6N1, for example, those don't match anything in the vaccine. Again, we're still seeing 100% survival in our mice and getting good results, both IN and IM. Next slide, please. We also did, this is the pathology and some transcriptomics. So what you're seeing up on the top is we saw a dramatic, significant reduction in virus titer in the lungs of these mice. When we did transcriptomics on the lungs here, you see the mock PBS column is all the way on the left. Those genes are all being turned on, lymphocyte activation, ROS response, programmed cell death, whereas you see much less activation in the vaccinated animals, both intramuscular and intranasal. And then the pathology, that left column, you see there's much more inflammation and pneumonia in the lungs of those animals compared to the vaccinated animals that looked quite good. Next slide. We also did similar experiments in ferrets. I'm not going to summarize all of this, but we did the same thing. We took ferrets, vaccinated them, and then administered, again, LD50s of virus. Next slide. And we saw similar results. We saw severe pneumonia and alveolar damage in the animals that were unvaccinated and very nice clean lungs in the vaccinated animals. And this was given, we did both intramuscular and intranasal. We're particularly interested in the intranasal, and the intranasal worked in all cases. Next slide, please. Okay, so that moves us on to the clinical trial. So once we had this product GMP manufactured, we had lots of nice data. We did a nice safety study in rabbits that gave us no difficulty. We were ready to begin this trial. So I designed a phase 1 clinical trial. And remember, it's a phase 1 study, so it's small. The goal is to identify any major safety signals that would happen in a large number of people, right? We're not going to find, small things that happen in 1 in 1,000, 1 in a million. We're going to find things that happen in, 1 in 10 people in this type of study. So the study is a 45-person study. We have 3 groups. One group received IM BPL-1357. One group is receiving intranasal BPL-1357, and one group is receiving placebo. Now, every patient, because it's a blinded study, is receiving a shot in the arm and sprays in the nose. We actually give the 1 ml in a divided spray, 0.5 in one nostril, 0.5 in the other, and then it's one shot in the arm. Everyone's receiving all of that and participating in the study. Next slide, please. And then this is the study schedule. So typical study, we're giving them a dose of vaccine 28 days apart, so they're getting 2 doses. We're not sure if 2 doses are necessary, but we're doing it so we can evaluate the immune response at both time points as well as safety. We're following everyone for 210 days, diary cards to collect adverse events as well as visits. I think the big key here, though, is that we're doing something that has not been done frequently in other studies, and it's something that I started doing in my challenge studies in the last few years prior to the COVID pandemic, which is we're collecting mucosal samples to look at mucosal immunity and what's happening after vaccination, whether it's intramuscular, intranasal, are we getting a mucosal immune response? And so we're using nasal absorption SAM strips, where these little strips you're seeing on the top that are placed in the nose. The patient holds their nose closed for about a minute. It collects secretions, so you can look for things like antibodies and cytokines. And then on day 7 post-vaccination, we're also, through the use of a nasal speculum, as you're seeing there, we're doing a cytology brushing of the middle turbinate to collect cells where we can do transcriptomics and other cellular assays to look at the cellular immune response. Next slide, please. These are the basic study objectives. Obviously, safety is a major objective here, assessing the acute safety of the vaccine. We're also quite interested in the immune response as well, to look at the immunogenicity to see if we're getting the same kind of responses that we saw that were protective in the animals. And then these tertiary objectives are actually the things that I'm quite interested in, really characterizing the immune response, because I think a lot that we learn from these types of studies is going to help us determine, is this vaccine where we want to be going? Does it have to be used in a certain way? For example, do we need 2 doses? Do we need multiple doses? Do we need to combine this with the other candidate and give doses of different combinations of viruses? Do we need to give a shot and an intranasal spray, or can we focus on just intranasal? There's a whole host of different things that sort of questions that could arise from really studying in detail the responses that we're getting from this vaccine and how we need to approach vaccines like this or universal vaccines in general for influenza. Next slide, please. So the strategy long-term here is, as I was mentioning, thinking about doses, routes, combinations of vaccines, additions of influenza B, dose de-escalation, what's right for kids, what's right for adults, what's right for people who have very little exposure to influenza, like a small child, versus someone who has years and years of history of exposure to influenza, like an elderly person. We'd like to do challenge studies in the phase 2 space in order to look at the ability of this type of vaccine to protect against infection in a challenge setting, as well as in the real world setting, where you need to do a much larger study, obviously, to look for protection. And then later on, we'd like to do challenge studies to look at breadth of protection. So, for example, I have a low-path avian H10 influenza virus that's under IND that could be used in a challenge study to test protection with this vaccine, an avian virus that would not cause severe disease or significant disease in humans, but could be used as a marker of breadth of protection. Now, in all of these types of studies, we think it's necessary to do careful analysis and comprehensive scientific exploration. That's what my blue arrows mean, is that I'm not just evaluating these vaccines in order to just say, does it work or not? I'm going to throw it away. I'm evaluating the vaccine to say, what does this do? Characterize what's going on, bring all that clinical data back into the lab, bring all those samples back into the lab, try to understand this so we can create new generations or just provide information that helps others who are working on these kinds of vaccines, and that's the goal. So the final update I have on this is we have 36 people enrolled and vaccinated so far. About 15 of them have received 2 doses, and we're looking to complete enrollment by November. Next slide, please. So thanks for having me today. I'm hopeful that we can move past where we were in 1918 with the government standard influenza depot, where they were giving whiskey, brandy, or stout as the treatment. To some degree, we haven't moved that far from there, but I hope we can. And I should say that, I thank the huge team of my own in the LID Clinical Studies Unit, as well as Jeff's team. And the study that I'm doing currently was funded by the Gates Foundation. Thank you.
Thank you, Dr. Romoli, and I want to thank both you and Dr. Osterholm for your presentations. We've got a question here in the room from Tim Cook.
Thank you for the overview. I wanted to ask a question. You probably hear it often, but where do you see the mRNAs kind of impacting the flu, whether it's universal or just better seasonal, or what are your thoughts on that? Obviously, it's early days for mRNA. Do you think it'll be able to be administered intranasally someday, or modified to get a mucosal response? I mean, where do you see that going?
So I think that mRNA is a platform, and there are many different platforms. I think the key is not really the mRNA. From my point of view, it's not the platform that's the most important. It's what's the antigen that you're delivering, right? So the mRNA is not the antigen. It's what the mRNA produces that's the antigen. And so you need to provide the right antigens in the right place in order to get protective immunity that's going to do what you want it to do, and whether that's protect against infection, protect against transmission, what have you. So whether or not mRNAs can be delivered intranasally, I don't know. I have some concerns about that, but it may be possible, and it may depend on, there's other parts of the platform, for example, the lipid nanoparticles and things like that. So I think it depends on what that is. But I think my big concern is that we keep taking the approach of, I'm going to find my favorite antigen that I think is going to induce the perfect immunity for flu, for coronavirus, for whatever it is. And I think there's a problem there, in that we're trying to sort of outsmart nature. And the reality is, when you develop immunity, you develop it to the whole virus. There's lots of different antigens there. There's the HA, there's the NA, there's the internal genes that induce T-cell immunity. I think we need to harness that. We need to think about, how can we create a very broad immune response? Because a child who's 3 and an 80-year-old woman and a 25-year-old with diabetes may not respond to the same antigen the same way. And I've seen this in some of my own studies. But offering different ways for them to respond, because there's different antigens present, I think is important. So whether or not mRNA can be used to do that kind of thing in the future and with the right sort of platform, I don't know. But I think we really need to focus less on the platforms and more on what kind of immunity we're trying to induce. And one of the big problems is we still don't really know for these kinds of viruses, and we need to learn more. And so I hope that everyone doing these kinds of studies tries to understand the immune responses they're getting and what's working and what's not, so that we can get that understanding.
Next question is from Dan Hoft on the line.
Thank you. Outstanding work, Dr. Memley. I'm just wondering if you could give us any more information on what you think your vaccine is doing mechanistically in terms of, I know it's a lot of different antigens and it's trying to get antibodies and T cells, but in the preclinical studies, do you have any evidence of one mechanism or another, or a combination?
Yeah, we do. I didn't have time to present all that data, and it's complex. But what you see in the preclinical studies is, when you look at viruses that have matching HA and NAs, you see a much more prominent role of neutralizing antibody, and oftentimes that's mucosal antibody. When you look at viruses that don't match well, so, for example, the H10, this is an H1, 3, 5, and 7, so there's no H10. Obviously, you don't see that neutralizing antibody as much, because we're not inducing neutralizing antibodies, and so you see a much more cellular response playing a role. So, but in all cases, everything's happening. It's just what seems more dominant in terms of offering protection. And obviously, these are the things we can measure, and there probably are things that we can't measure yet from the mucosal samples or even from the systemic samples. So we do see some mechanisms. Some of that is summarized in the paper, so you could look at the paper. I don't really have time to get into all of it now, but we are doing that. And in addition, I should mention we're currently moving and engaged in a preclinical study to look at transmission blocking as well with these vaccines in ferrets. So that's ongoing work.
Thank you.
Dr. Ostrom, any comments you want to make at this point? You're on mute, I believe, sir. Dr. Ostrom, I think you're on mute still.
Oh, sorry about that. I was on mute this side. I just wanted to say that I very much appreciated Matt's comments in the previous question about the mRNA, and I very strongly agree with him on that, and think that the point that he made is really the critical issue about which vaccines will or will not work. It's much less about the platform and much more about what, as he said, the antigen.
Well, I want to thank both of you for your presentations today. Fascinating, and we look forward to hearing more as we go forward. I think all of us are going to be very interested in watching these correlates of protection and looking for durable immunity as we improve influenza protection among our human population.
Thanks.
Our next presentations are focused in a bit different direction. Our next panel is vaccines and reduced risk of dementia. There are several vaccines being investigated as a potential tool to reduce risk for developing Alzheimer's disease. Currently, there are more than 55 million people living with Alzheimer's and other dementias, and 10 million new cases diagnosed every year worldwide. In this session, we'll hear from Dr. Paul Schultz, a professor and vice chair in the Department of Neurology at the University of Texas, Dr. Stephen Lehrer, a physician at Mount Sinai, and Dr. Peter Reinstein, a geriatric medicine specialist at Severna Park, Maryland, Dr. Avi Berkbinder, a resident physician at Mass General Hospital, and Dr. Svetlana Ukrainetsa, an associate research professor at Duke University. And I apologize to those whose names I may have incorrectly pronounced. You have the floor, sir.
Thank you very much. It's a real honor to be here today. Paul Schultz from UT in Houston. The previous two talks were fantastic and set the stage for talking about vaccination affecting the risk of Alzheimer's disease, a totally different direction. Next slide, please, from what we've been talking about today. Next. Oh, okay. That's even better. Thank you so much. I'm going to make 3 points today. One is that the immune system is complex. Multiple items that activate the immune system make Alzheimer's worse. And finally, we're going to talk about whether another form of immune system activation, vaccines, is helpful or harmful for Alzheimer's patients. I feel like I'm bringing coals to Newcastle. You guys know the immune system more than I do. Many different cells involved. As a neurologist, it's stunning how many are involved. And the complexity of the system, it's even more complex, as you know, because they all talk to each other and they release factors that go throughout the body and can affect the immune system anywhere. So point number one is that the immune system is very complex, and there's a reason why I'm mentioning that. And certainly I don't understand many nuances, but I'm not even sure every immunologist knows all the nuances of it yet. The immune system is activated in Alzheimer's disease in a very interesting way. As you know, the hallmark of Alzheimer's disease is amyloid plaques accumulating in the brain of people who eventually develop symptoms. The amyloid comes from a normal brain protein that is incompletely metabolized, and it's very chemically sticky. So if one molecule that's partially broken down sees another one that's partially broken down, they stick to each other, and over decades, you develop amyloid plaques. We think that people have the plaques for up to at least 20 years before they come into our clinic with their very first symptom. Now, the important thing for today is that around every amyloid plaque is activated microglia. So the immune system is very active. We can see it on biopsy and autopsy, but also now I have PET imaging agents that will allow us to see this. And we can see that in one person with Alzheimer's, there's hot areas, so to speak, where the microglia are very activated. Differs by person, but everybody has the immune system activation there. Now, if a pathogen gets into the nervous system, bacteria, virus, the things being released by these microglia are not a bad deal, right? They would kill the bacteria. They might kill a few brain cells, but the person won't die of meningitis or encephalitis. In this case, though, we think that the microglia are activated for decades, and they're accidentally killing brain cells that whole time until enough are gone that a person comes into clinic and the wife says, Paul's memory is not as good as it used to be, okay? So a very important role of the immune system, we think, in Alzheimer's disease at baseline, okay? Now, the second important fact to know is that everything that we see in clinic that activates the immune system is bad for Alzheimer's, okay? Someone comes in with a urinary tract infection, or they call The wife calls and says, my husband's gotten a lot worse this week. And we find a urinary tract infection or pneumonia, or they have a broken bone. Anything that activates the immune system, even though it's in the periphery, seems to do something in the brain to accelerate Alzheimer's disease. We think what it's doing is releasing factors in the periphery that activate the central inflammatory marker processes more than they already are and accelerate the disease. A lot of times then, after the urinary tract infection is over, a person doesn't go back to where they were. They're now different. They've accelerated. They've got 6 months more to the Alzheimer's disease in the space of just a week, okay? So, second message then is that the immune system is active in Alzheimer's disease. It becomes more active under certain circumstances and seems to make people with Alzheimer's a lot worse. So up until this last year, when patients and families would ask me about vaccination, what I would say is that, well, I would tell them that the flu is deadly, shingles is a problem, pneumococcal pneumonia is deadly, get the vaccine. But in my own mind, I was thinking the vaccine is going to activate the immune system just like anything else that I see in clinic every day activates it. I assumed it probably made things a little bit worse, but it was worth it because it would save the person's life. Well, it turns out I was totally wrong with that hypothesis, thank goodness. And the focus today is going to be on the fact that, yes, there's a lot of things that activate the immune system and make Alzheimer's worse, but we have several vaccines you'll hear about today that actually seem to be protective against Alzheimer's disease for quite a few years. It's even more complicated because different vaccines have different degrees of reduction in risk for Alzheimer's. And sitting and listening to the previous two talks as a neurologist, I was thinking, there's obviously not one influenza vaccine. There's many. They work through different mechanisms. They have different routes of entry and so forth. So it's not surprising that there's a lot of variability in the outcome. But nonetheless, many of us are finding the same general outcome, which is that vaccines can be helpful for Alzheimer's to varying degree. And with that, I'll turn it over to our next speaker. Thank you.
Dr. Lair, you have the floor.
Yes. I'm just going to start the video, okay. Today I'm going to talk about shingles vaccination that reduces the risk of both Alzheimer's disease and Parkinson's disease. And my co-author, Peter Reinstein, is actually there right now in the hall. Let's see. Your camera covers. Yeah, I mean, my camera is on. I don't know why. Wait a minute. Wait, I'll try again. Join meeting. Wait a minute. Hang on. Control. Join. Wait a minute. Let's see what's going on here. Meeting passcode. Yeah. Wait a minute. Join.
I think it's the aim of your camera, sir. We've seen your hand. Now I see your face in the corner of the video. Dr. Reinstein, why don't you go ahead, and then we'll hopefully get Dr. Lair to catch up with us.
Okay, I could do that, but maybe let me say to Steve, we can hear you perfectly fine. We can see the slides. So why don't you go ahead and present? Not worry that we can't get a close-up of your face. Steve, are you there? Okay, I guess I will go ahead. First of all, I want to thank you for inviting me back here to the Humphrey Building. I used to work at Food and Drug Administration for 25 years, and I spent lots of time in this building. What Dr. Lair and I have been working on is looking at new tools that have become available. particularly the UK Biobank, its ability to correlate events in a patient's life, like receiving a particular vaccine, with conditions later on. There are any number of new statistical tools available, and we've built up quite a library of them. We'll be happy to discuss those. But let me tell you, why don't we go to the first slide? And what we're saying is that, and we heard it already before, infectious disease. Oh, sorry about that. Oh, okay, and I have a clicker and I can advance my own slides. Infectious diseases might confer a risk of Alzheimer's disease, and protection against bacterial and viral infections is beneficial to the brain. In a study looking at flu, 5.1% of flu vaccinated patients and 8.5% of flu unvaccinated patients developed Alzheimer's disease during a 46-month follow-up. That's quite a significant difference. Being vaccinated against pneumonia between ages 65 and 75 was associated with a reduction in the risk of Alzheimer's disease afterwards. And again, it was a very significant reduction. Let's see, we'll make it over to the next slide. It may be that herpes simplex type 1 and herpes zoster virus interact to trigger Alzheimer's disease. May be that herpes zoster actually activates the dormant herpes simplex type 1 virus. And herpes type 1 virus appears to be implicated in the development of Alzheimer's. What does HSV-1 do? It may produce low-grade inflammaging, depending on how you want to pronounce it. It's a relatively new word, so I guess it's open to variation in pronunciation. And finally, beta amyloid in Alzheimer's may be well, it's for sure an antimicrobial, and it may be an inherited disease against herpes simplex virus. What Steve Lehrer and I found was a 15% dementia risk reduction in vaccinated subjects. Our findings suggest that the shingles vaccination reduces the risk of dementia, and it's consistent with the link between the viruses and Alzheimer's disease. Might mention that obviously the vaccine we were looking for was the older vaccine, Zostavax, because of the time period in which the vaccines were administered. We're going to need to do it again when there's enough patients accumulated to look at the effects of Shingrix. We assume it's going to do the same thing. And Schneer found reduced dementia incidence after varicella vaccination in Wales. So the unanswered question then is whether the reductions in the incidence of Alzheimer's will be additive. And that's something we'd like to look at in the near future. We've also got some evidence that shingles vaccine reduces the risk of Parkinson's disease. What this is, each of these dots represents one state in the United States. Sorry about that. This is the slide I want to have up there. Each dot represents one state in the United States. So what you're seeing here is the proportion of adults age 60 and over who ever received a vaccine. And then what you're seeing is it looks like it's going in the wrong direction, but this is the rank of the state. So a state that has the lowest incidence of Alzheimer's actually gets the highest rank. So the best states are down here, and the better states are over in the place where they've got the highest the highest percentage of Alzheimer's vaccine. Get over to the next one. I did. Again, shingles vaccine reduces the risk of Alzheimer's. I'm looking at the wrong slide. We've gotten a hypothesis as to why all of this might be true, and I want to spend just a minute or two on that. Dr. Lair and I feel that Alzheimer's disease and Parkinson's disease may be due to embryologic reactivation of processes and pathways silenced at birth. We're thinking that this neuroinflammation may remove the methylation, the other things in the tails of the proteins applied to the genes, and that once this is done, this process is irreversible. So medicines or vaccinations that undo this process, well, they can't undo this process. So the neuroinflammation, the removal of the acetylation, those are permanent, and essentially what was developed during embryologic stage is destroyed late in life with this activation. Got some interesting correlations with fetal alcohol syndrome, which we can talk about if there are questions. And, you know, that's sort of what I wanted to present, or what Steve wanted to present. Steve, are you there?
Yeah, Peter, I am, but can you hear me?
We can hear you just fine.
Oh, good. Well, yeah, you've done so. So go to the next slide then. If you go to the next. Okay, here's the AD and PD don't spread willy-nilly through the brain. They progress in a predictable fashion. The Braak stages, AD and PD, represent a complex physiologic process gone awry. And the advanced Braak stages 5 and 6 are almost identical in AD and PD. And in their advanced stages, Alzheimer's and Parkinson's are, I've learned this from Peter, who handles, who's certified in geriatric medicine, among other things, that in their advanced stages, Alzheimer's disease and Parkinson's disease are difficult to distinguish clinically from one another. Go to the next slide. As you can see, here are the Braak stages. To the left is Alzheimer's, and the right is Parkinson's. And if you look at Braak stages 5 and 6 in both Alzheimer's on the left bottom and left upper, and Parkinson's on the right, you'll see the similarity of the regions of the brain that are involved. That basically agree with Peter's observation that the end stages of both Parkinson's and Alzheimer's look quite similar. You can't tell them apart. Next slide. So there are various characteristics that you could explain of Alzheimer's by the embryologic activation. The NSAIDs lessen the risk of Alzheimer's but are ineffective as a therapy because the NSAIDs suppress inflammation, but they're not a cure because they can't silence the embryonic genes that become active and are causing the brain damage. And then children with fetal alcohol syndrome have abnormal performance in learning and memory activities. But light to moderate wine drinking appears to lessen the risk of dementia and cognitive decline. So alcohol, disrupts the embryology of a fetus's developing brain, lowering IQ, but alcohol interferes with embryologic reactivation in the elderly, lowering the chance of Alzheimer's. And then apparently low-dose radiation treatment, it looks like it could be beneficial in patients with Alzheimer's disease, but ionizing radiation has a significant negative impact on the growing brain. And the inconsistent association between herpes viruses and Alzheimer's disease could be because herpes viruses are not a direct cause of the disease. It's likely that it's the embryologic reactivation and gene unsilencing is increased by herpes viruses. Next slide. Gene silencing and reactivation, as Peter's already mentioned, they're controlled by histones. The nucleus of every cell has 2 meters of DNA to create chromatin nucleosomes made up of 8 histone proteins are first wrapped around the DNA. And then the tails of the histones carry chemical modifications such as methylation, acetylation, and ubiquitination. And these numerous proteins have evolved to read and write and remove these histone marks. Next slide. No, back. You got to go back one. Advancing age is the greatest risk for AD, and why is that? It's because these chemical modifications of the histones, the methylation, acetylation, ubiquitination, silence the genes, but these modifications are not bolted on with iron bands. The chemical forces hold them in place until the end of reproductive life. Thereafter, evolution is indifferent as to what happens to them. With advancing age, the chemical modifications are dislodged by viruses, infections, and inflammaging, and embryologic pathways and processes silenced at birth are reactivated. Alzheimer's and Parkinson's and other forms of neurodegeneration are the result. And cancer is actually an exact analog of this because cancer is also a disease of aging. And many cancers, the common cancers such as colorectal and lung, they develop after an accumulation of genetic mutations that occur with age. Now, next slide. And once, as Peter mentioned before, once the embryologic reactivation occurs, it's unstoppable. And it relentlessly destroys what it created in utero. Next slide. And here's a summary. The flu and pneumonia vaccination reduced the risk of Alzheimer's. Herpes simplex type 1 and herpes type 2, herpes zoster may interact to trigger Alzheimer's. The shingles vaccine Zostavax reduces the risk of Alzheimer's by 16% and to 20%, and also Parkinson's. But the unanswered question is, are the risk-reducing effects of flu, pneumonia, and shingles vaccination on Alzheimer's, are they cumulative? Finally, Alzheimer's and Parkinson's may be due to embryologic reactivation of processes silenced at birth. Next slide. All of the slides in this presentation are available from our Fermata Pharma website. If you go to fermatapharma.com, you can see all of the slides. And next slide. Here, there are the references that you'll get to see, and that's what I have to say.
And if you want to see what Dr. Lehrer looks like, there's a great picture of him on Wikipedia. So just Google Stephen Lehrer Wikipedia and you'll see him.
Thank you.
Thank you very much, Dr. Lehrer and Dr. Reinstein. Our next presenter is Dr. Avi Berkbinder, a resident physician at Massachusetts General Hospital. Dr. Berkbinder, you have the floor.
Thank you. Thank you for having me. So, as said, I am a resident at Mass Gen, here to talk about the risk of Alzheimer's after influenza vaccination, a recent publication in the Journal of Alzheimer's Disease. So, some table setting for today. I'll start by talking about kind of the background, which I think has already mostly been done, but talking about the issues with this kind of investigation, some of the potential biases that we encounter and need to address. Then present some of the published results, and then I'll also present some forthcoming unpublished analyses, the results of those that I think will be interesting. So to start with the hypothesis, influenza vaccination, as discussed, alters the effects or alters the risk of Alzheimer's disease. At least that was the question that we were exploring. The problems that you encounter with this kind of question are risks of confounding by indication. So, for instance, someone who has COPD may be more likely to see their doctor. More healthcare exposure might be more likely to get the jab, right? To get influenza vaccinations or some other vaccine. Alternatively, people who have high adherence to healthy lifestyles, who have things like good exercise habits, might be more likely also to get the influenza vaccination. And we know that those healthy lifestyles might be protective against Alzheimer's risk. So ultimately, the approach that we took was something called propensity score matching, the PSM, and I'll get into that in a little more detail in a few slides. Our study population was 1.8 million people, half of which had the influenza vaccination and half who did not. So the data source was the Optum Clinformatics Data Mart, which is a commercially available product from Optum or United Health that institutions can subscribe to. We use data spanning from 2009 to 2019. We specifically stopped before the onset of the coronavirus pandemic in the United States to avoid confounding. All of our subjects were at least 65 years old at the start of follow-up. And in this case, when I say follow-up, we divided our 10-year period into two periods. So one look-back period, which ranged from 2 to 6 years, depending on the specific analysis, and one follow-up period that ranged from 4 to 8 years, again, depending on the analysis. The look-back period is to identify good study candidates and measure covariates, and the follow-up period is essentially the observation period. Notably, the Optum Healthcare or the Optum Clinformatics is only consistent of people with insurance, both medical and prescription. It's both Medicare Advantage, private, and Medicare Advantage with Part D. And of course, as part of the look-back period, we ensured that we excluded anyone who had mild cognitive impairment, dementia, or any sort of encephalopathies, as well as dementia medication, so any indication that they had dementia. So let's refer back to what I mentioned a moment ago about the issues we encounter with this kind of study, the healthy vaccinee bias or the confounding by indication. So briefly, just to give a primer on propensity score matching, this is a way to create essentially two cohorts where the main difference between them is the vaccination status. So in a randomized controlled trial, you ideally have the probability of exposure is 0.5 if there's two arms. That's the random component. This is essentially a method for pseudo-randomization. So what you do is you take all of these covariates and you come up with a propensity score based on the covariates. The propensity score is the predicted probability of exposure, in this case, vaccination. Then you assign every person in the cohort or in the sample a propensity score. So that is their predicted probability for being vaccinated. And then you match people who were in reality vaccinated and in reality were not vaccinated based on their propensity score. There have been a number of studies showing that the balancing effect of this method is quite robust, and that it provides if done well and verified afterwards that it's well balanced, provides a good estimate of the true effect. And so we included some of the things that we balanced on. So things like demographics, including where people were from. So what region of the United States, since there are significant variabilities in vaccination uptake. Things like how often they saw the doctor, and how many routine annual examinations they had in the period. Comorbidities like COPD, things that we know might be related to either vaccination or Alzheimer's. And medications, things like benzodiazepine use, anticholinergic use, etc. So the results. So I think Dr. Reinstein actually mentioned the percentages from the study a moment ago in the prior presentation. So you can see in the two-way frequency table that there's about a 35,000 difference in the number of people who developed Alzheimer's disease between those who were vaccinated and those who were not vaccinated. You'll notice that the row-wise totals are the same, since it's a one-to-one matching. So it's about a little over 900,000 for the vaccinated cohort and the unvaccinated cohort. The relative risk, therefore, was 0.6, meaning that people who were vaccinated at least once with an influenza vaccine were 40% less likely to develop onset or be diagnosed with Alzheimer's in the 4-year follow-up period. The absolute risk reduction also was 0.034, and I provide the number needed to treat there. So I mentioned that we had some variable look-back period durations and therefore follow-up period durations. The slide I just showed you was for the 6-year look-back and 4-year follow-up. 6 years of look-back is quite conservative. But we did some follow-up analyses saying, what if we made the duration longer? So the bottom 2 rows, you'll see that the duration of the look-back is 2 years, and therefore the duration of the follow-up is 8. And you'll see again that the effect sizes, although a little bit smaller, are still quite, remarkable. The other question we came across, or we thought of, was what if we only looked at people who are older than 75? So again, we see an effect size. In this case, it's actually larger than than the initial question of over 65. So this slide is largely illustrative. This is based on a secondary analysis, a time to event analysis that is not as rigorous as the previously discussed propensity score matching algorithm. But I include it, 1, because it provides a very clear illustration of the effect, and 2, because it allows us to do some analysis of whether there's a dose-response association here. And we see that when we look at the 6-year look-back period, people who received 6 vaccinations actually had a lower risk of Alzheimer's disease, or lower hazard rate of Alzheimer's disease compared with those who received 3. So those were the studies, or those were the results of the recently published article. Now I'm going to present some results of some follow-up analyses that we've been working on. So we've spoken a lot today about adjuvants, conjugation, various types of vaccines. So one question we had was, is there a difference in the protective effect between the adjuvanted vaccine on the market, Fluad, and the non-adjuvanted? And indeed there is. The relative risk when you compare those who received the adjuvanted vaccine, Fluad, versus those who received non-adjuvanted standard dose vaccines was 0.46. We had just done this analysis when I prepared these slides, but the absolute risk reduction was about 0.017. So not as strong of an effect as the initial analysis, suggesting that the initial vaccination really provides the most effect. And this is just a kind of a small additive bonus. And then the other question we had was, there's a recent CDC approval, or rather encouragement, that people 65 and older receive the high-dose Fluzone vaccination. And so our question was, is there an association between the dosage of the flu vaccine and the effects that we see on risk of Alzheimer's disease. And again, there is. You'll see that if you compare the Fluzone high dose versus Fluzone standard dose, the relative risk is 0.79. So people who received the high dose were about 20% less likely to develop Alzheimer's disease compared to those who received the standard dose Fluzone. And if we compare the high dose Fluzone to any standard dose influenza vaccination, the effect size is a little bit larger, 0.74. So this is moving away from influenza for a moment. We also looked at a few other routine adult vaccinations. So on this first row, you'll see zosters or shingles. This is primarily Zostavax, since we only had about 2 years of the Shingrix approval during our study period. And you'll see that it's not quite as strong as the effect size we saw for the influenza vaccination, but we did see that there was a reduced risk of Alzheimer's following shingles vaccine. Now, notably, we didn't control for shingles exposure in this study. That's a follow-up component that we're working on currently. So I think it's important to provide that caveat. Then Tdap is another one that we looked at, and this had a similar effect size. And finally, the pneumococcus vaccines, I think, are perhaps some of the more interesting ones. The PCV13, the conjugated one, had quite a strong effect size, similar to the Tdap shingles influenza. But the purely polysaccharide version, the PPSV23, while still providing some reduced risk of Alzheimer's, some protection, was not nearly as strong as the others. So I think that provides a window into the possible mechanistic components going here. That's all I had. I think there's an interesting point to be made that is not from our studies, but there's a number of studies looking at BCG vaccination, particularly for patients with bladder cancer. And as you all know, these patients sometimes receive intravesicular BCG immunotherapy. So it's essentially put onto the walls of the bladder. And again, there we see that there are people who received the BCG vaccine or BCG immunotherapy had a reduced risk of Alzheimer's disease compared to those with bladder cancer who did not. Those studies are out of the US and Israel. And so I think these are places where the tuberculosis load the burden is not very high. So again, I think that provides us with a little more insight into the possible mechanisms, and that this may not be so much of a protection against infection and the inflammation resulting from that infection, although I certainly think that's a component. But I do think there's some other kind of component of perhaps epigenetic reprogramming or innate training going on here. Thank you.
Thank you, Dr. Berkbinder. Our next presenter is Dr. Svetlana Ukrantseva, an associate research professor at Duke University. Dr. Ukrantseva, your slides are up. You have the floor.
I'm Svetlana Ukrantseva from Duke University, working on connection between Alzheimer's and aging and Alzheimer's and infections and vaccines. And I will talk about repurposing of existing vaccines for Alzheimer's prevention, including our results and some mechanisms. Please, next slide. To understand better why vaccines can be beneficial for Alzheimer's, we should touch role of infections in Alzheimer's disease a little bit, and I will do it, and then I will present our findings from our group about pneumonia and shingles vaccines as candidates for repurposing for Alzheimer's disease prevention, and then concluding remark with some challenging questions. Next slide, please. So, what is role of infections in Alzheimer's disease? Are we talking of some specific pathogens or more like compromised immunity? Very different pathogens, viral, bacterial, fungal, have been all linked to Alzheimer's disease and related traits across studies of different nature. It's human, animal, some cell cultures, etc. You can see quite a few references about 6,000 references with which are discussing or somehow connecting Alzheimer's and infection is up to date. So this indicates the possibility that reason for this connection may be not only specific microbe, but rather compromised immunity, which may increase brain's vulnerability to various pathogens and through this contribute to neurodegeneration. And this is actually very well in line with what we currently know about major Alzheimer's disease risk factors, such as aging, which is strongest risk factor, certain genes and exposures, because many such factors may affect immunity and the brain vulnerability to infection. Next slide, please. So, this is just an example, and I will focus on aging due to time limitation. So aging, at the bottom, you see aging, some exposures, and genetic risk factors, like major risk factors for Alzheimer's disease. And you know very well that aging is characterized by immune senescence, and this means several things, like thymus shrinkage, slower responses to antigens, decline in blood-brain barrier integrity, and all this contributes to slower immune response and decline in immune surveillance. And this means that some viruses, bacteria, anything which is residing in the body may start to reactivate, which the best probably zoster, varicella zoster, which sits in dorsal ganglia for decades and very well controlled by immune system. But as we age, immune surveillance weakens, and this virus starts moving up to the brain and contributes to damage, relevant damage, and to neurodegeneration. So what aging does through immune senescence, it promotes increase in burden of microbes and their products in the brain and relevant damage. Other factors such as exposures and genetic risk factors relevant to Alzheimer's disease also contributes to decline in immune surveillance, increase in blood-brain barrier permeability, and to reduce ability to repair respective damage. Especially it refers to genetic risk factors such as ApoE4, which altogether like increase in burden of infection-related damage and decrease in ability to repair this damage may contribute to neurodegeneration. So Alzheimer's disease-related risk factors, major risk factors, could be major contributor to this connection between infection and Alzheimer's disease. Next slide, please. So if compromised immunity does play a role in Alzheimer's disease development, then we would expect that vaccines, some vaccines, not all, with broad beneficial effects on immunity might be protective against Alzheimer's disease. And growing evidence suggests that some vaccines indeed show beneficial off-target effects on health and survival traits that are far beyond the protection against specific disease. And these vaccines are pneumonia, flu, against pneumonia, flu, shingles, tuberculosis, BCG, and there is quite a few references up to date. It's just some examples of these references here, which show that such effects do exist. And it gives us some hope that such vaccines have also potential to be repurposed for Alzheimer's prevention due to their broad beneficial off-target effects on immunity. And I should say about mechanisms of such effects because, frankly, few mechanisms are discussed, maybe many more, but mostly discussed mechanisms involve trained immunity, such as macrophages, innate immunity, and producing lymphocytes with cross-antigens reactivity. Also, there was discussion that it might be direct effect of elimination or reducing incidence of disease, which these vaccines work for, such as, for instance, you don't have pneumonia, you don't have probably relevant damage to the brain, and so it decreases the risk. However, several studies, like here shown, like Schneer et al. 2022 and Scherer et al. 2021, they separated effects of reduction of disease from effect of vaccine. And concluded that protective effects of vaccines on Alzheimer's disease and more generally on dementia, they are not due to elimination of target disease. So mechanism can be other than I just mentioned, it, like trained immunity and cross-reactivity of lymphocyte, but it is area of current investigation and far from be finished. Next slide, please. I will show our findings from our group about pneumococcal and shingles vaccines as candidates for repurposing for Alzheimer's disease prevention. Next slide, please. It's results from health and retirement study using data on about 6,000 of HRS participants linked to Medicare records and ICD codes, and using propensity score matching techniques, which was just discussed. I will not spend time on it. You already heard. We estimated associations of herpes simplex, zoster, shingles, pneumonia, and recurrent fungal infections diagnosed between ages 65 and 75, as well as vaccinations against shingles and pneumonia received between these ages 65 and 75 with onsets of Alzheimer's disease and Alzheimer's disease combined with other dementias, as well as with all-cause mortality risk, to see how it compares. Next slide, please. This is a brief summary of the results, and the main point is the focus on this yellow column, is that we can see that all these infectious diseases, viral, bacterial, and fungal, viral it's herpes, bacterial pneumonia, and fungal mycosis, they all increased risk of Alzheimer's disease. And both vaccines against shingles and pneumonia decreased this risk. What we can also see here, that when we combine Alzheimer's disease with other dementia, it's a second column, gray area, gray column. The effect diminishes, with exception of mycosis, fungal infection. But generally, it becomes less strong, less pronounced, less significant, despite the fact that sample size of such, say, combined group Alzheimer's disease and other dementia is twice larger than just for Alzheimer's disease. It's 1,500 versus 750. with Alzheimer's disease, which is indication that Alzheimer's disease connection to compromised immunity and so effects of infections and vaccines is maybe more specific for Alzheimer's disease than for other dementia. So it's maybe feature of Alzheimer's disease, this connection. Next slide, please. So what we see from this health and retirement study results, as I said, is different infections all associated with Alzheimer's risk. Both vaccines lowered the risk of Alzheimer's disease, and associations were stronger for Alzheimer's disease alone than for Alzheimer's disease combined with other dementia. Next slide, please. And in cardiovascular health study, we looked on how interaction between vaccine and genotype affects Alzheimer's disease, and not just any genotype, but we focused on gene which is involved in both Alzheimer's disease and vulnerability to infection, to see if it may modulate effect of vaccine on Alzheimer's disease. Next slide, please. It's a complicated table, but it's a very simple idea here. If we do not do any stratification, it's part one, we can see that effect of pneumonia vaccine and number of vaccination with and without accompanying flu shot has a sort of moderate effect on Alzheimer's onset after age 75, which is highlighted in yellow here. However, when we stratify by genotype, by carriers and non-carriers of Alzheimer's disease risk factor, which is here variant of SNP RS 6859 in Nectin-2 gene. And this gene is very interesting. We found it earlier to be both Alzheimer's disease risk factor and involved in host response to infections. And other researchers also published this connection. So in carriers of risk factor for Alzheimer's disease, the effect becomes much more pronounced and significant. We see a larger reduction in risk and more significance, and the effect disappears in non-carriers of Alzheimer's disease risk factor. But for other SNP, I should say, it could be different situation. We're talking only about this one. I'm not generalizing here. So next slide, please. So as summary, it's basically line in the middle. Pneumococcal polysaccharide vaccine, which was this vaccine, may provide genotype-specific protection against Alzheimer's disease, and so it could be promising finding indicating that personalized prevention of Alzheimer's disease, taking into account risk factors, genetic risk factors for Alzheimer's disease, may actually work. Next slide, please. Next, concluding remarks. So, I would say that our results, as well as results of other groups, generally support the idea that Alzheimer's may be linked to compromised immunity rather than to specific microbes. And using this heterologous, aka non-specific, aka off-target vaccines could be beneficial for Alzheimer's prevention. Our own results support the opposing of pneumonia and shingles vaccines for Alzheimer's prevention, and also show that genetic risk factors for Alzheimer's disease may modulate the effect of vaccines on Alzheimer's, providing opportunity for personalized prevention. However, we should always remember that, for instance, we talked about shingles vaccines. It was Zostavax live attenuated virus vaccine, and beneficial effects of inactivated vaccines, such as Shingrix, need confirmation because off-target effects of vaccines might be different for live and non-live vaccines. And next slide follows challenging questions. So much more research is needed to clarify these challenging questions. First, how many of the existing vaccines could provide the off-target benefits for Alzheimer's? It seems that all vaccines may do this because many vaccines show such effects. However, some studies show that some vaccines have negative off-target effects on all-cause mortality. And this is like elephant in the room. So these vaccines still protect against target disease, but have reduced survival, which is not good at all. And it's not quite clear why. So it's need to be clarified before we saying vaccine is beneficial for Alzheimer's. Let's repurpose this vaccine. But if it negatively affects all-cause mortality, it could be trade-off, and we need to address this trade-off. So one explanation of such negative effects refers to this live versus non-live vaccines. I mean, it's still discussed in the literature that live vaccines may be more beneficial than non-live vaccines. But this needs confirmation and further research because it's mostly anecdotal evidence. Even some vaccines like TDP showed such effects in children. Like live vaccines are better, but it needs further research. Next slide. That's basically it. It's acknowledgement to my biodemography of aging research unit, our collaborators, NIH grants, of course, and both studies. Thank you so much.
Thank you, Dr. Rukhain-Seva. Are there any questions in the room or online? Rob Schechter?
Thank you very much for the presentations. I was wondering, in your examination of specific vaccines, whether you've had a chance to look at any effect of receipt of multiple vaccines along with individual vaccines, if there's any additional benefit from receipt of multiple vaccines. Second question is, you've implied that there's a point of no return at which vaccination is unlikely to be helpful, and wondering if that point is before or after initial diagnosis of Alzheimer's, and whether there'd be any perceived possible benefit in slowing progression or reducing severity by immunization at time of diagnosis.
We looked only on effect of vaccines applied before diagnosis, so as risk factors. But we are going to look after diagnosis as well. It may be completely different, results. And about direct effect of elimination disease due to direct effect of vaccines, as I probably mentioned, other studies showed that you cannot explain protective effect of vaccines, off-target effect of vaccines on dementia just by the effect on target disease. So it's probably different mechanisms, such as trained immunity or else. So it needs further investigation. I cannot hear.
Dan Hoft has a question online, and he'll be our last question for this panel. Thank you.
Thank you, Bob. And great presentations from all of you. Thank you very much. I just wanted to follow up on what had already been presented about it's with outcomes types research. It's always hard to know for sure if you've ruled out any other variables. And prospective trials are critical, of course, to really prove an effect. at least in most cases. So I wonder, is there a way to do a prospective trial here, especially with the ethical dilemma of withholding a vaccine to see in a control group, for example?
Well, one of the ways of addressing that is going to the animal model. We have a very well accepted animal models of Alzheimer's disease and Parkinson's too. And because the animals are raised together, we can rule out whether getting the infection or not is an issue. We can actually give them the infection too to study infection versus vaccine. I think that's going to be the way to go because, as you said, I think it'd be real tough in humans with a potentially deadly virus like influenza to withhold vaccines from humans, whereas the mice don't complain.
Well, I want to thank the members of this panel. It's been very interesting and a different perspective than how I normally think about our vaccines. Is I want to get them into anybody and everybody at all times, but I've now got another lever for some of my older patients who may have concerns. Thank you. We have reached the end of the time for this panel. We're running over just a bit. Let's take about a 5-minute break and we'll come back at 3:10 to start our next panel, a somber milestone, COVID-19 claims more than 1 million Americans. Thank you.
Produced by the U.S. Department of Health and Human Services.